The origin of microfluidics is intricately tied to the rapid advancement of the semiconductor and microelectronics industries during the mid-20th century. The sophisticated microfabrication techniques developed during this era, specifically photolithography and wet chemical etching, provided the fundamental manufacturing infrastructure necessary to manipulate solid materials at the micrometer scale. Initially, these subtractive manufacturing processes were engineered exclusively for the creation of integrated circuits, focusing on the precise patterning of silicon wafers to direct electrical currents. However, researchers soon recognized that the same lithographic techniques utilized to etch electrical pathways could be creatively repurposed to carve physical topographical conduits for fluids. This conceptual leap marked the transition from solid-state microelectronics to microelectromechanical systems, eventually leading to the specialized discipline of microfluidics. The undisputed milestone of this foundational era was achieved in 1979 by S.C. Terry, J.H. German, and J.B. Angel at Stanford University, who successfully developed what is widely considered the first true lab-on-a-chip. Their pioneering work demonstrated the fabrication of a miniaturized gas chromatograph integrated entirely onto a silicon wafer. Utilizing standard semiconductor photolithography and chemical etching, the research team constructed a functional analytical system that allowed for massive size reductions compared to the conventional benchtop laboratory instrumentation of the time. The silicon-based device incorporated an integrated injection valve and a remarkably long 1.5-meter capillary separation column coiled intricately into the microscopic footprint of the substrate. A thermal conductivity detector, manufactured in separate batches, was integrally mounted onto the wafer to monitor the output. The performance of this early device was exceptional for its size, demonstrating the ability to separate complex gaseous hydrocarbon mixtures in fewer than 10 seconds. Despite the evident success and revolutionary nature of Terry's gas chromatograph, the anticipated explosion of microfluidic technology did not immediately materialize. Instead, the field experienced a pronounced 10-year-long gap in development and commercialization. Several critical factors contributed to this stagnation. Physically, the performance of these early etched silicon channels was hindered by their geometric limitations. The etching of crystalline silicon typically resulted in rectangular or trapezoidal cross-sections, rather than the perfectly circular profiles of conventional drawn glass capillaries. These non-circular symmetric separation channels introduced severe mass transport limitations, leading to significant analyte band broadening and ultimately lowering the peak capacity and overall separation efficiency of the analytical device. Furthermore, the commercial translation of this early technology proved exceedingly difficult. Although companies like Microsensor Technology, Inc. were founded in 1981 to capitalize on these miniaturized gas chromatographs for portable ambient air quality monitoring and planetary probe applications, the broader industrial ecosystem required to support large-scale microfluidic manufacturing was entirely absent. Fragmentation in application demands and a lack of standardized macroscopic interfaces meant that the technology struggled to secure a lucrative foothold. It was not until 1990 when William R. Higdon purchased Microsensor Technology, Inc. on the verge of bankruptcy and consolidated its sensor and silicon microvalve production that the fundamentals of commercial microchromatography were firmly established. Consequently, the 1980s remained a period of dormant potential, awaiting a conceptual framework that could adequately bridge the gap between microscopic engineering and analytical chemistry. The modern era of microfluidics was officially catalyzed in 1990 by the seminal theoretical work of Andreas Mons, N. Graeber, and H. M. Widmer. Recognizing the limitations of simply miniaturizing individual sensors, Mons and his colleagues introduced the concept of the Micro Total Analysis System, µTAS. Their conceptual framework was revolutionary because it shifted the focus from merely shrinking a single detector to miniaturizing and integrating the entire analytical process, encompassing sample preparation, injection, reaction, separation, and detection onto a single contiguous microchip. The core of Mons's argument relied heavily on the fundamental scaling laws of physics and fluid dynamics. Theoretical models demonstrated that as the characteristic dimensions of a system are reduced, the physical behaviors of fluids change in highly advantageous ways. For instance, a decrease in the characteristic length exponentially decreases the volume of reagents required, slashing costs, increasing analysis speed, and reducing the generation of hazardous waste. More importantly, miniaturization drastically alters mass transport phenomena. Because diffusion time is proportional to the square of the distance across which molecules must travel, shrinking a channel to the micrometer scale allows diffusional mixing and chemical reactions to occur in mere fractions of a second. Mons's argument utilized dimensionless parameters to predict these behaviors, illustrating how the Fourier number, describing the ratio of diffusive transport to the rate of storage, and the Bodenstein number, characterizing the relative importance of convective flow to axial diffusion, scale favorably in microenvironments. This scaling advantage meant that µTAS devices were inherently faster and theoretically capable of higher resolution than their microscopic counterparts. The practical realization of the µTAS concept found its first major success in the integration of capillary electrophoresis onto planar microchips, spearheaded by researchers such as D.J. Harrison and J.M. Ramsey. Capillary electrophoresis separates charged molecules based on their electrophoretic mobility within an applied electric field. Microfluidic environments proved exceptionally well suited for this process because the high surface-to-volume ratio of microchannels facilitated rapid heat dissipation, allowing for the application of much higher electric fields without causing sample-degrading thermal runaway. Additionally, the use of electrokinetic flow eliminated the need for complex, miniaturized mechanical pumps, as the fluid was propelled purely by electrical potentials. However, this era was heavily defined by the ongoing struggle to identify optimal substrate materials. Silicon, the inherited material from the semiconductor industry, quickly revealed its severe limitations in chemical and biological analyses. Silicon is opaque to visible and ultraviolet light, which effectively precluded the use of standard optical detection methods such as fluorescence microscopy. Furthermore, silicon is electrically semiconducting, which proved highly problematic for capillary electrophoresis, as the extreme voltages applied across the channels could lead to electrical breakdown or short-circuiting unless thick, robust insulating layers were meticulously deposited. To circumvent these electronic and optical issues, researchers transitioned to glass substrates. Glass provided a suite of essential advantages. It is highly optically transparent, electrically insulating, chemically inert, and exhibits a very low inherent fluorescent background. Its electrical properties made it the perfect substrate for supporting the high electric fields required for microchip capillary electrophoresis, and its transparency allowed for seamless optical monitoring of moving analyte bands. Despite these benefits, glass was notoriously difficult and expensive to manufacture. Etching deep microchannels into glass required handling highly dangerous chemicals like hydrofluoric acid, and sealing the channels required anodic bonding, a process demanding extreme cleanliness, perfectly flat topography, high temperatures, and high voltages. The fragility, high fabrication cost, and bonding complexity of glass substrates meant that rapid prototyping was virtually impossible, restricting microfluidic research to well-funded laboratories equipped with specialized cleanroom facilities. The field urgently required a material paradigm shift to democratize fabrication. The technological bottleneck imposed by silicon and glass was decisively shattered in 1998 with the introduction of soft lithography and the widespread adoption of polydimethyl silicone. Pioneered by George M. Whitesides and his research group at Harvard University, this paradigm shift democratized microfluidics, transforming it from a highly specialized, expensive engineering discipline into an accessible tool for chemists and biologists worldwide. PDMS is a silicon-based organic polymer that transitions from a viscous liquid to a transparent, rubbery elastomer upon curing with a cross-linking agent. Soft lithography decoupled the design iteration phase from the cleanroom. Instead of directly etching every single microchip, researchers could utilize photolithography just once to create a durable, reusable master mold, typically made of a photoresist patterned on a silicon wafer or chrome-coated glass. Liquid PDMS was then poured over this master mold, cured thermally, and peeled away, instantly replicating the microscale and nanoscale topographical features with astonishing fidelity. This replica molding technique reduced the fabrication time from days to mere hours and slashed costs significantly. Beyond manufacturability, PDMS possessed inherent material properties that were revolutionary for biological applications. It is highly optically transparent down to the ultraviolet spectrum, chemically stable, non-toxic, and crucially, highly permeable to gases, allowing living cells cultured within enclosed microchannels to continuously receive oxygen and expel carbon dioxide. The elastic nature of PDMS also catalyzed the next major leap in the field, the development of active fluidic control. In the year 2000, Stephen Quake's group introduced multi-layer soft lithography, a technique that exploited the elastomeric deformability of PDMS to create the first robust monolithic microvalves. By casting two separate layers of PDMS with differing ratios of polymer base to crosslinker, the layers could be bonded irreversibly into a single monolithic structure, completely avoiding the interlayer adhesion failures common in rigid micromachining. The Quake valve design featured a pneumatic control channel positioned directly above or below a fluid flow channel, separated by a remarkably thin PDMS membrane. When pressure was applied to the control channel, the thin membrane deflected into the flow channel, sealing it completely, with response times on the order of milliseconds. Because these pneumatic actuators relied on the inherent softness of the material rather than complex micro-machined silicon gears, they could be miniaturized to an unprecedented degree. This capability enabled the era of large-scale integration in microfluidics. Quake and colleagues successfully packed thousands of these microscopic valves into highly dense combinatorial arrays, operating them via multiplexers. Much like the binary logic architecture that drives electronic integrated circuits, fluidic multiplexers permitted the complex routing of fluids across hundreds of individually addressable reaction chambers, using only a minimal number of external pneumatic control lines. Specifically, a multiplexer operating as a binary tree allowed the control of numerous independent fluid channels with only a logarithmic number of control inputs, directly enabling high-throughput screening applications where single cells or molecules could be isolated and analyzed in parallel with unprecedented precision. This period witnessed the emergence of multi-phase droplet-based microfluidics, driven by the need to eliminate Taylor dispersion, a phenomenon where fluid velocity gradients cause solutes to spread and dilute along a continuous channel. Researchers such as Rustem Ismailov, David Weitz, and Stephen Quake explored the manipulation of immiscible fluids. By forcing aqueous solutions and inert fluorocarbon oils to meet at specific microfluidic geometries, researchers generated continuous streams of highly monodisperse water in oil droplets. These tiny droplets, often picoliters in volume, functioned as self-contained independent microreactors. Encapsulating biochemical reactions within these droplets isolated them from the channel walls, completely eliminating cross-contamination and surface adsorption issues.
This methodology fundamentally altered biochemical assays, enabling ultra-high throughput directed evolution, digital polymerase chain reaction or dPCR, and single-cell transcriptomics. By successfully linking genotype and phenotype within a confined picoliter droplet, the capacity to rapidly screen millions of enzymatic reactions became a tangible reality. As microfluidic platforms matured and stabilized physically, the discipline experienced a profound thematic shift. The primary focus evolved from the rapid separation of chemical analytes to the complex cultivation and interrogation of living biological systems. Standard two-dimensional cell cultures in macroscopic petri dishes had long been criticized for their inability to accurately predict human physiological responses, primarily because they failed to replicate the intricate three-dimensional architecture, fluid shear stresses, and biochemical gradients characteristic of living tissues. Microfluidics, capable of establishing steady-state concentration gradients and continuous perfusion at a microscale level, provided the exact spatial and temporal control necessary to mimic the true cellular microenvironment. This biological pivot culminated in the advent of organ-on-a-chip technology. The seminal breakthrough in this domain occurred in 2010 when Donald Ingber and his team at the Weiss Institute at Harvard University unveiled the lung-on-a-chip. This biomimetic microsystem revolutionized in vitro modeling by proving that physical forces are just as critical as chemical cues in governing tissue behavior. The device consisted of a flexible, porous PDMS membrane that compartmentalized two parallel microchannels. Human alveolar epithelial cells were cultured on one side of the membrane and exposed to air, while human pulmonary microvascular endothelial cells were cultured on the opposite side and continuously perfused with a blood-like culture medium. The true ingenuity of Ingber's device lay in its recreation of dynamic mechanical forces. By applying cyclical suction to adjacent vacuum chambers, the flexible central membrane was periodically stretched and relaxed, precisely mimicking the cyclic mechanical strain of human breathing. The introduction of mechanical breathing led to profound physiological discoveries. When the device was exposed to environmental nanoparticles or pathogenic bacteria, such as E. coli, introduced into the air channel, the mechanical stretching exponentially enhanced the absorption of nanoparticles across the barrier and triggered a robust inflammatory response. Endothelial cells activated the simulated immune system, prompting circulating white blood cells to adhere to the membrane, undergo diapedesis to cross the barrier and engulf the invasive bacteria. This ability to visualize and quantify complex, integrated organ-level responses without relying on animal models offered an unparalleled mechanism for drug toxicity testing and disease modeling, paving the way for the replacement of expensive and often inaccurate mammalian test subjects. Simultaneously, the quest to translate microfluidic capabilities into the clinical sphere drove the maturation of centrifugal microfluidics, commonly referred to as lab-on-a-CD platforms. While PDMS chips often required a tangled array of external syringe pumps, pneumatic controllers, and tubings, a major impediment to deployment in resource-limited clinical settings, centrifugal platforms cleverly bypassed these macroscopic constraints. By integrating microchannels and reservoirs onto a compact disk substrate, researchers utilized the pseudo-forces generated in a rotating reference frame to manipulate liquids. When the disk spun, the radially outward centrifugal force pumped the fluid from the inner radius toward the periphery. By precisely controlling the angular velocity and designing specific geometric barriers or hydrophobic constrictions, researchers could dictate the sequential bursting of valves, enabling complex fluidic operations such as aliquoting, decanting, and stoichiometric mixing. Because the microfluidic channels were narrow, the flow remained strictly laminar. characterized by Reynolds numbers well below the turbulent transition threshold, which necessitated specialized designs to induce chaotic advection for fluid mixing. Furthermore, centrifugal platforms inherently facilitated density-based separation. Whole blood could easily be fractionated into plasma and cellular components simply by rotating the disc, completely bypassing the need for separate sample preparation steps. Because the entire fluidic propulsion mechanism relied entirely on a single, inexpensive rotary motor, lab-on-a-CD technology rapidly became a dominant architecture for automated immunoassays and point-of-care (POC) clinical diagnostics. While traditional polymer and centrifugal microfluidics advanced the frontiers of biological research, their reliance on clean room fabrication, continuous cold chain reagent storage, and expensive optical readout systems largely restricted their use to modern hospitals and affluent academic laboratories. To address the severe global disparities in healthcare, particularly in the developing world, the focus shifted towards extreme affordability and true democratization. This initiative was spearheaded by the development of microfluidic paper-based analytical devices, µPads, introduced by Andres Y. Martinez and George M. Whitesides in 2007. Moving away from elastomers and plastics, the researchers utilized standard cellulose filter paper, such as chromatography paper, as the foundational substrate. By patterning the paper with hydrophobic barriers, utilizing methods such as wax screen printing or PDMS plotting, they created distinct hydrophilic microchannels capable of guiding fluids in complex 2- and 3-dimensional networks. The utilization of paper was transformative due to its inherent physical properties. The porous cellulose fiber network automatically draws fluids through the channels via capillary action, entirely eliminating the need for external power sources, mechanical pumps, or centrifuges. Paper is extremely lightweight, easily transportable, biocompatible, and completely disposable via simple incineration. Because a single µPad could be manufactured for mere fractions of a cent, it perfectly aligned with the World Health Organization's assured criteria for diagnostics designed for developing nations. Assays were typically designed to yield colorimetric results, allowing a user to diagnose conditions such as liver function or glucose levels simply by observing a visual color change on the paper surface. Despite the theoretical elegance of these diagnostic platforms, the broader commercialization of microfluidics faced a notorious valley of death, spanning the enormous gap between academic prototyping and successful market penetration. The most significant engineering hurdle was the world-to-chip problem. Academic prototypes often functioned flawlessly on a stabilized microscope stage, but required highly trained technicians to operate delicate macroscopic interfaces, including specialized tubing, microscopic connectors, and complex pressure controllers. When transitioning from a controlled laboratory setting to a chaotic clinical environment, these fragile interfaces frequently failed, leaked, or suffered from catastrophic dead volumes. The microfluidic devices that successfully crossed this valley were those that completely abstracted the fluidic complexity away from the end user. The Abbott iStat system emerged as the quintessential success story of this era. The iStat circumvented the world-to-chip problem by integrating all necessary fluidics, chemical reagents, and biosensors into a single and closed disposable cartridge. A clinician needed only to place a minute sample of blood as low as 17 microliters into the cartridge and insert it into a rugged handheld analyzer. The device automatically executed pre-programmed fluidic manipulations over chemically sensitive silicon biosensors to perform electrochemical detection of blood gases, electrolytes, and cardiac markers within an average turnaround time of just 2 minutes. By internalizing the microfluidic complexity and relying on standardized, foolproof cartridges, the iStat revolutionized emergency, critical, and rural point-of-care diagnostics. This era also witnessed the critical realization that the lack of universal hardware standards severely hampered modularity and commercial growth. In response, organizations began drafting harmonization guidelines, culminating in standards like ISO 22916. This international standard specified basic interoperability requirements, standardized pitch spacing dimensions for microfluidic port connections, and provided classification frameworks to ensure different microfluidic components and macroscopic interfaces could connect seamlessly, reducing the reliance on proprietary, incompatible hardware. Furthermore, to remove the final barrier of expensive readout instrumentation, developers began leveraging the ubiquitous presence of the smartphone. Modern smartphones possess advanced optical sensors, immense processing power, and global connectivity. By developing physical add-on lenses or dedicated software applications, the smartphone camera effectively replaced bulky laboratory microscopes, analyzing the colorimetric outputs of paper µTAS or the fluorescent signals of microdroplets in real time. This integration allowed instantaneous data transmission to remote healthcare databases, cementing the reality of decentralized, highly connected diagnostic networks in resource-limited settings. As microfluidics enters its modern paradigm, the field is undergoing a fundamental transformation in manufacturing methodologies, biological complexity, and computational design, driven by the convergence of additive manufacturing, artificial intelligence, and a heightened global demand for rapid diagnostics. Historically, the replication of microfluidic channels was bound by the two-dimensional nature of photolithography and the subsequent layer-by-layer bonding required by PDMS soft lithography. Fabricating complex, three-dimensional overlapping channel architectures using PDMS is highly labor-intensive, plagued by optical alignment errors and prone to delamination under high fluidic pressures. Today, high-resolution 3D printing technologies are actively challenging PDMS as the gold standard for rapid prototyping. Technologies such as stereolithography (SLA), digital light processing (DLP), projection microstereolithography (PUSL), and two-photon polymerization (2PP) have achieved unprecedented microscale and nanoscale resolutions. Additive manufacturing constructs the entire microfluidic device sequentially by curing photosensitive resins layer by layer with ultraviolet lasers or light projectors. This subtracts the need for expensive master molds and eliminates the bonding step entirely, yielding monolithic, robust chips capable of housing complex internal three-dimensional matrices, helical mixers, and interwoven vascular architectures that are physically impossible to mold using traditional soft lithography. Simultaneously, biological applications have evolved from isolated organ models to fully integrated human-on-a-chip systems. Recognizing that systemic human diseases and drug toxicities are rarely confined to a single tissue, researchers have begun fluidically coupling multiple distinct organ-on-a-chip modules to simulate comprehensive whole-body pharmacokinetics and pharmacodynamics. A prominent configuration in this domain is the gut-liver-lung axis. In these highly complex microphysiological systems, an orally administered pharmaceutical compound is first introduced to the intestinal epithelial module, where its initial absorption and potential efflux are modeled. The permeated drug is then transported via vascular microchannels to the hepatic module, closely mimicking the physiological portal vein circulation. Within the liver compartment, cultured hepatocytes perform first-pass metabolism, potentially altering the drug into its active therapeutic metabolites or generating toxic byproducts. For example, research utilizing these platforms has successfully tracked the liver metabolism of the prodrug capecitabine into its active form 5-fluorouracil. These metabolites are subsequently routed to a downstream target organ, such as the lung or heart, allowing researchers to evaluate secondary toxicities and therapeutic efficacies.
This sophisticated multi-organ integration captures the intricate interplay of the human systemic response, providing an exceptionally predictive preclinical platform that far exceeds the physiological relevance of traditional in vitro assays. The immense structural and operational complexity of these networks has outpaced human intuition, prompting the extensive integration of artificial intelligence and machine learning into microfluidic design and real-time operation. Historically, optimizing a microfluidic channel geometry to achieve a specific fluid dynamic outcome, such as perfect fluid mixing at low Reynolds numbers, required computationally expensive FEM simulations combined with tedious iterative trial-and-error prototyping. Today, AI algorithms execute advanced inverse design methodologies. By training convolutional neural networks and utilizing Bayesian optimization on vast datasets of fluid behaviors, the AI can independently predict the necessary structural parameters required to achieve a target flow profile. A researcher simply inputs the desired performance metric, such as a specific droplet size, generation rate, or mixing efficiency, and the algorithm automatically computes the optimal geometric boundaries. Tools like the DAFD web platform automate droplet generator design, circumventing weeks of manual numerical simulation and resulting in highly counterintuitive but functionally superior architectures. Furthermore, within operational devices, machine learning algorithms continuously analyze real-time image data from integrated sensors, facilitating automated droplet sorting, cell tracking, and dynamic feedback control of microvalves to rectify blockages or optimize reaction conditions instantaneously without human intervention. Finally, the trajectory of microfluidic technology was permanently altered and massively accelerated by the COVID-19 pandemic. The acute global necessity for rapid decentralized viral detection shattered historical regulatory inertia and forced the immediate deployment of sophisticated point-of-care microfluidics. Technologies that had been gestating slowly in developmental pipelines were thrust to the forefront of global healthcare. The Cepheid Gene Expert System, utilizing automated self-contained microfluidic cartridges to perform reverse transcription polymerase chain reaction (RT-PCR), received some of the earliest emergency use authorizations, enabling hospitals to obtain highly accurate molecular results in just 45 minutes rather than waiting days for central laboratory processing. The pandemic also drove molecular diagnostics directly into the consumer market. Lucira Health successfully commercialized the first FDA-authorized over-the-counter molecular home test for COVID-19 and influenza. Their disposable device leveraged advanced microfluidics to automate a reverse transcription loop-mediated isothermal amplification assay, reading the resulting pH-induced color change via integrated optoelectronics and microprocessors. Although Lucira faced complex financial and regulatory hurdles that ultimately culminated in bankruptcy just days before their combination respiratory tests were fully authorized, the technological precedent was unequivocally set. The pandemic conclusively proved that microfluidic platforms could be reliably scaled and mass manufactured, effectively overcoming the commercial valley of death to provide critical lab-quality diagnostic capabilities directly at the point of need. From the rudimentary silicon gas chromatographer developed at Stanford to today's AI-optimized 3D-printed multi-organ systems, microfluidics has fundamentally reconstructed the architectural boundaries of chemistry and biology. By continually mastering the unique physics of the microscale, the discipline has transformed from a specialized analytical curiosity into an indispensable pillar of modern medical and biomedical engineering.